Hello and welcome to the Best Practices Summit. Pollinator Friendly Alliance is a grassroots 501c3 nonprofit that protects the natural world through the conservation of pollinators. Our co-host is the Xerces Society, a science-based nonprofit.
Okay, hello everybody. My name is Anne Marie Journey, as you can see. I am several kinds of entomologist. I've worked with the University of Minnesota in agricultural entomology. I worked with the Minnesota Department of Agriculture doing invasive species monitoring in pine forests and pine stands. And I've worked with now in my 15th year with Hennepin County with the Wetland Health Evaluation Program. So I do aquatic entomology as well. And the soil part of all of this kind of gets going with my PhD research. I did biological control of corn rootworm for my PhD. This is way back in the Stone Ages before we had GMO corn. I was using a nematode, an entomopathogenic nematode, so it carries with it a bacteria that once released into the insect's blood space by the nematode kills and digests the insect, allowing the nematode to swim around for a while in a soup of dying insect or dead insect. Really a bad way to go. I don't recommend it to anybody. And after that, I participated in some research in the pre-release stages of GMO corn. So first with the Roundup Ready corn and then with the stacked corn. So my work kind of had me in cornfields, and I thought about this earlier listening to Dr. Lundgren. During that transition from old style, either soil applied or broadcast insecticides that you put on in pounds per acre, through GMOs, and then into the introduction of neonicotinoids when we're talking about ounces per acre. And during that whole time, the thing that I noticed, and it became more obvious to me recently, going back and looking over some of my notes, was that there were fewer things other than corn living in cornfields during this whole period. And it went from corn, and then there would be other plants, especially at the field edges. The first 4 or 5 rows would be a competition between corn and the things that were growing outside of the field. A lot of that used to be milkweed, to corn, just corn. And then there would be some weeds around the edges of the field, but the field edges tended to get smaller during this period as well, because you could go farther and farther out with your crop, closer and closer to the edge of the field, because you were going to kill everything else anyways, so it really didn't matter. And I saw fewer lacewings in the field eating the aphids. I saw fewer ladybugs in the field. I saw fewer spiders in the field. I saw fewer pollinators buzzing overhead. It just kind of became simpler and simpler and simpler, more and more and more like a factory floor. So as we describe commercial or industrial agriculture, what we're really describing is a progressive simplification of that ecosystem to just the plant you want and not much else. And that has had a lot of implications that probably were not fully appreciated at the time, because the whole idea was about production, getting the biggest yield, the most out of the acre. And as Dr. Lundgren noted, farms that are doing regenerative agriculture are actually more profitable because they're not putting in all of those additional inputs. After my time with the University of Minnesota, I contracted for 4 years with Minnesota NRCS as the ACES Soil Health Coordinator. And I was involved with a project called the Minnesota Soil Health Initiative. And this is when I got educated about soil in a way that I would have helped a lot if I had been when I was doing my PhD work. because I learned that the reason I couldn't come up with the basic, perfect, optimal combination of nematode species and rate of nematode applied to get the desired result, which was, of course, getting rid of the corn rootworms, was that I was working in a conventional field and the soil was just incredibly unhealthy. And I know it was now because now I know what soil health looks like. And I learned that doing the Minnesota Soil Health Initiative. Pretty simple idea. NRCS contracted with farmers to plant at least 2 species of cover crops for at least 5 years in the same spot and not till in the fall. That's all. Very simple. Usually farmers were using 3 species. Sometimes we'd go in and find 4 because there was carryover from the prior year. And then in 2016, my colleagues and I went out and we started sampling the fields, the cover cropped fields, and then a closely paired non-cover cropped field. And we did 36 different tests, some in the field, some in the lab on those 2 environments. And we had trouble telling them apart. There were some things that, as it turns out, we could say were very different. Just 5 so far have held up to statistics. So the number of earthworms in a cover crop field versus the non-cover crop, the smell of the soil temperature, the percent bare ground. All of this should be fairly intuitive. One field has plants growing in it, the other field may not. And then the extracellular enzymes that we can recover from soil that fungi in particular put out to acquire carbon when they are breaking organic matter down and recycling it into the soil. Almost significant was infiltration rates. And what we found going through, if you'll pardon me, switching my notes here a moment, we found that when we looked at initial survey data from these farms, that farmers had already seen that. And they'd seen things that were a lot more complicated than we could measure with our big, effort, bulk density and water holding capacity, and send soil down to the lab and get the chemical analysis done on the soil. They were seeing the things that were multi-layered in their complexity. But the power of observation was there, and it was there within about a year of starting to grow cover crops that farmers were noting this. So when Lori and I were talking this last year about what she might like to see me do for soil health, instead of telling you what soil health is, or telling you how you should promote soil health, and it's the big 5, so reduce disturbance, keep your soil covered. I hope you can't hear my dog, she's going off. Have more plants growing on the land, have more plants growing on the land longer, so maximize your growing season, and then return herbivores where practical, keeping in mind that herbivores are in all sizes, from like elephants down to small insects. Rather than just talk about that, what she thought would be interesting, and what I thought would be interesting as well, would be to give you an idea of how you might look at the soil yourself, how you might put the power of observation to work for yourself, whether you live in a city. I have to tell you that a potted plant is not in soil. So this evaluation will not work for your potted plant because it's in medium. But if you have even a small yard like I do here in Minneapolis, you can take a soil and shovel rather, and go outside, take a look at your soil and get an understanding of what's going on with it with just a few simple tests. So last fall, beautiful day, Lori and I went out and we went to a place where there were a lot of different soil types available. This is from Web Soil Survey. Each one of those orange lines, if you can see it, is a different soil map unit, and just sat down and went at it. So shall we go outside? Start the video. So the first thing I'm encountering is a rock, which I had to work my way past. And then I want to go all the way down with my poacher spade. And I'm trying to pull out an intact block of soil, and that came up so beautifully. I'm not going to mess with it from there. Okay, here we have a fairly intact block of soil. I'm using a data sheet that we put together for a dynamic soil property sampling project, and it kind of runs through all of our field assessments for soil health. So the first thing I did is I used my handy thermometer to take an air temperature in the shade when we started. That was 61.9. I have since jammed it down as far as it would go, and it's already rising. It was 53.2. So at about 4 inches, the soil was 53.2, and this is harder when you're doing a partial. At 2 inches, it is, I gotta get this written down before I forget it, 0.2 at 2 inches, we have 53.6 when it stabilized. So just your basic kitchen meat thermometer, nothing fancy. In fact, a lot of the stuff that I brought with me is out of my kitchen. So I brought a strainer, a tea strainer, and a little glass bowl, and I've got some water. And this is a kind of a miniature slake test. I'm going to take some of this soil. I'll get some from right about there. Here's the soil. Put it in the tea strainer. It's a very crumbly soil, Lori. And so we'll see. I'm going to let it wet on its own. So you just take your handy dessert dish that you get your creme brulee in from the grocery store. Very handy things, these. And put your tea strainer down in, and then set it aside, because you're going to let it wet on its own. You don't want to agitate it. So then the other thing I'm going to do is get my measuring tape so that that's handy. Obviously, I'm not going to need additional help in breaking up this soil for the VES, or the visual examination of soil structure, because it is beautiful and it's already kind of breaking apart on its own. So I'm not going to need tools to help me break it. I have my handy magnifying glass because I want to take a look at what might be living in this soil. So I have a couple of petri dishes. You don't need to use petri dishes. I just happen to have them at home because I'm a bug geek. And you can use leftover containers. Those are great. So this is basically your kind of little soil lab. If you got a couple of little jars around, there's something you really want to look at under better magnification or better light than you have out in the field, you just put it in a jar. So we're going to start with the soil itself as it came out of the field. I'm not going to pick it up too much because it's going to want to break on me pretty easily. The surface of the soil, you can see it right here, has a good organic layer. So we've got all these moss, we've got grasses, we've got little bits of leaves in there. So there is an O horizon on the soil. It's not necessarily thick, but it's there. The other thing to note is right here, when I pulled this out of the ground, it broke. So that's a horizontal fracture. In the VES, we would do this part and this part separately. But when I look at it, tell me a little bit about the history of this space. Was this area changed in about the last 80 years and how it was managed?
There were no trees here. There was a barn just south of here, and there were cattle.
Okay, so we might be seeing the old condition and the newer condition reflected in that break. So with the vest, what you do is you just break it apart with your hands. And you look at things as you go. So I'm moving on, having started with that part of my data sheet. Horizontal fracturing observed. Indeed. And it was observed at about 3 inches. So that usually means that there was a compacted layer, and I think that compacted layer is probably about 3 inches down. Now I'm going to take a smell. I've kind of let it out in the air for a few minutes, but I'm going to come in here where it's still intact, take some, and smell. And down here it has what we call a weakly earthy smell. Okay, so I've got that earthiness, and you usually end up with literally a brown nose when you're doing this in the field. But I'm also going to come up here where it's livelier. See all those roots?
Yeah.
That are in there? Now smell that.
Oh, yeah.
Yeah, much different. So here we have a lighter textured soil. It feels like it's a little sandier than what we've got up here. There's more organic matter, I believe, right here, and certainly more active organic matter. So that's one of the things I'm going to note on our soil smell scale. It's a 3-point scale. So 1, you smell nothing, or you smell something chemical or sour. If you smell hydrogen sulfide, rotten eggs, that means the soil is saturated all the time. You can smell, not here you wouldn't get it, but on some fields you can smell the chemicals that are in the soil. That's a 1. 2 means that it's lightly earthy. 3 means it's richly earthy. Now, when we were out doing this one study, the Soil Health Initiative, I think we only had 2 or 3 instances out of 44 fields where somebody awarded a 3. And we usually do this by consensus scoring because smell is so subjective. On this one, that's pretty richly earthy. I suppose I have brown lips. Generally. This is the risk of doing this work on camera.
You look good.
If I have to get out my mirror, tell me. I don't know if I have one on me. This one I would probably give like a 2.5. There's a little room because it's not as dark a soil to go above a 2. But the bottom half, where we first smelled it and it was lighter, it was harder to pick up that scent, that would be a 2. So I'm making that note, 2, 2.5 top. Then I'm going to look, as I break it down, for horizontal roots. So the vest structure means I need to take this and do it separately. And did you see how easily that just came apart? All I did was turn it, and this top layer with all the roots stayed together, and everything else is down there. So, but there are good roots down here too. If you look, the plants have been doing their work, especially the fibrous roots of the grasses, which is why I wanted to get into a grassier area to dig it. Now, that's beautiful. That's just gorgeous. That just falls apart. I don't have to hurt myself to work with this soil. This comes apart really easily. So when we look at this, it would be aggregates readily crumble with fingers on the scale. But it doesn't go to dust. It easily crumbles with fingers, but you can see that it doesn't go to dust. And there, and this would be one I want to take a picture of on my camera, is the rhizosphere in action. So you have, can you see it?
Yep.
Okay. You have the aggregates that are actually on the root. So looking at the table here, I would say that this matches up pretty well in the middle. and this is something that the University of Illinois put together, which would mean it was somewhere between 2 and 3% organic matter. It could be up to 4% because it's kind of right on the border between these two colors. This is easier to do under bright light than it is under dappled. It is not in the 3.5 to 7% range of organic matter. Organic matter is everything that is alive or was ever alive in your soil. It's what makes it brown. So the darker the soil, the more the organic matter. Okay, so the next thing that we're looking at, I'm just going to refer to my other handy thing pretty quickly. So we've done smell, I haven't seen any earthworms yet, but this is an area with a lot of trees and we sometimes see fewer. I'm looking for soil life as I'm going. I just pointed out the rhizosheath where the soil is being held together by the roots. We've done organic matter, so in a lot of the basic tests that we teach people at soil and water conservation districts and at NRCS field offices, we've done a lot of our biological assessment already. But let's really break this soil down and see what we get as we're breaking it. So this bottom part, still just nothing big. It comes apart just so easily in your hand, and yet as I'm looking at it, there are all kinds of little roots holding it together. I wonder if that's the rock I hit with the shovel. This is really nice brown cottage cheese texture soil. Let me get some that's right against the shovel. So another thing you can do, oh, here's an older root in there. You can kind of figure out how much clay is in the soil. This is a nice moisture. By texturing, I am not going to make this all muddy to texture it. I could. I'm not that good at texturing. What I am better at is making pinch pots, because I do ceramics. And I cannot get this to form a ball enough for me to even think about putting my thumb in it. So this is probably a fairly low clay soil, but it has that little gritty feeling on my hand, so I know that it has some sand in it as well. If I were to get my hand wet, well, I might as well. Let's get muddy. Then I would probably find that there's some silt in it. So you can feel all the 3 mineral components of soil. The sand, you can tell, gritty. The silt is what makes soil, when you do this, have a feeling of almost butteriness. So this feels really quite sandy here, and the clay is not too much. There is some silt in it, I can feel it, but not a whole lot. So this is a great soil for trees and probably for backyard gardens, but it wouldn't have been good for farming, or at least not that good, not by Minnesota standards. So still looking at what I'm finding, I have an old tree root. Take a look and see if anybody is walking along it. This root has been attacked by shredders, which is an important thing to note in the soils, and it also has some active fungi growing on it. Down there, the white things are the hyphae of the soil, of the fungi rather. And I know that there have been arthropods working on it because they have gotten between, is that a good spot?
Yep.
They have gotten between the bark and the inner connective tissue, and they've been working in that space. So there are a lot of beetle larvae and other arthropods that like to work in that space. This is the first stage in our return to the soil of the nutrients from organic matter. If there isn't an invertebrate there, a mite, a worm, an insect, to start attacking that stored carbon and breaking it down into smaller pieces, then the bacteria and the fungi that we're asking to do the rest of the job are slowed down because they work on surfaces. So if you see right here, there's a surface. It's been roughed up. It's dark colored, and there's that fungal hyphae growing on it.
because there's more surface right there for the fungus to colonize. And if you don't have that roughing up, that breaking down, that just chewing stuff and spitting it out that all of our arthropods and our other invertebrates are doing in the soil, then you make the whole aspect of breaking this stuff down and recycling the nutrients that much more difficult for the bacteria and the fungi to do, because they are surface dwellers. Fungi, once they've colonized, can run their hyphae into something. But that first colonization is on a surface. Now let's, because this looks pretty moist, let's take a look at this. This is called the sink strainer test. So you can do this at home with a tea thing or a sink strainer. I've let this soil soak. And we're going to see how well it kind of holds together now that it's wet. I'm just going to tip that over. And you can see it held together. So it is wet. It's wet all the way through. But it's not falling apart. The roots, the little rhizomes of the root and the soil granules being bound together, they're holding it together. There's enough of the organic matter from the rhizosphere, from everybody that's living here, to hold these granules together. This is not going to fall apart in a rainstorm. Also, you see that not very much of the fine components of the soil just fell off of it and landed in the water. So if you think of soil as having 6 components, and the 6 being sand, silt, clay, air, water, and organic matter, in a good soil, typically half of the space, half of the volume of the soil is air and water. And so when water got access to this particular soil, the soil held together just fine. The air space was replaced with water. I then turned it out. That's a fairly violent thing to do to it. But it held together. Now I can break it up, but it's wet all the way through. So this is a very well glued together soil. It has good structure. If I put it in an agitator and I had various sieves of different sizes, I could sort its components, but there would still be some aggregates that were pretty big in a soil like this. So that is excellent. It is healthy in that regard. Now let's keep breaking this up so that I can generate the all-important best score. When you have a soil that breaks on a layer like this, you do each layer separately. So far as I'm looking, and I've got my glasses on so I can see small things, I have seen some mites moving, but they are really fast. I have not come across any earthworm tunnels yet. I have discovered an old ceramic. This is one of the things about working in soil. You do have to be careful because that's really sharp. Part of a mark on it. Dinner plate, maybe?
Yeah, they used to bury their garbage years ago.
Okay, yeah, I've got a spot like that in my backyard in Minneapolis. So I won't put that back in the ground. Still seeing this beautiful crumbling, no problems. I haven't seen any horizontal roots, which can be a sign of compaction. The fact that there was a layer is interesting. It means there was compaction at some point, but I'm not hitting the platy structure that really compacted soil has. Let me get into a little different position here and see if I can get me any critters. The soil invertebrates, and by the way, this would be an SQ1 on the scale, very, very easy to break apart in that layer. But the invertebrates are going to be, there we go, up a little bit by the roots. So there we have, right there, you can see it, little worm in there. Earlier I saw a couple of beetles that were running around in here, but they've gotten away from me. Probably because they know I killed so many of their kind doing my PhD. So just looking. Now, here's another thing. Because this is a mossy area, you've essentially got on the top here this carpet of the moss that has just held the top together. The moss and the grass roots. Down.
There.
So they're just completely holding it together. The filamentous roots of the grass, here we can see the taproot of the dandelion. Dandelions are, by the way, actually perennial. So these roots are going to keep going as long as they can into the winter. So right now, all of these fibrous and tap roots, and then the structures of the mosses, because they are still green and they are going, they are catching sunlight and they are pumping root exudates into this soil. So they are making themselves a good carpet backing. Sorry about that, Lord. And in that carpet backing, if you want to think of it that way, is where I'm seeing more very small worms. I've got some mites, I'm pretty sure. Let's see if I can tell. Not too much that's going to show up without a microscope, unfortunately, but I could take some of it home because I do have microscopes and then take a picture for later consumption of some of the things that are living in here. So when we look at soil and we're looking for things that are living in it, and we focus on earthworms, which is the easy thing, you can always see them, you know what you're finding. And unfortunately now increasingly in Minnesota you're at risk of finding an invasive species with Asian jumping worm. We're ignoring the vast bulk of the invertebrates that live in soil. So in a given acre you might have 110, 120 million arthropods, and about 80 million of them would be mites. And they are really, really hard to see bare-eyed or even with a hand lens because they're so small. So right now I could probably be looking at a few thousand of them and just not recognizing them because they are so tiny. I think I've seen some movement once or twice, which is kind of how you pick them up. Got some glass in here too. So I guess I picked the trash pit pretty well. So because this soil has aggregates that readily crumble with your hands, most of them there's a size element to this, mostly under 6 millimeters when crumbling. So I do have, let me get some, do have my ruler in here. So for size, you can see how small the aggregates are. So mostly under 6 millimeters, highly porous, yes, roots throughout the soil, soil breaking up into these fine aggregates. The action of breaking the block is large enough to reveal natural or reduced fragments of less than 1.5 centimeters in diameter. Yes, right there, hanging on a root.
Yep.
Okay. You've got some small stones in here. I would give this soil on the VES score a 1. The description of that is friable. That's very much what this soil is. So let me see, what else would I say? I would give it the 1. I looked for I have not seen earthworms, but I only did this one. If we were going to do an earthworm count, I would be digging up a 1 square foot, or 1 cubic foot rather, block of soil. That's the unit for earthworms. So not worrying about that. I am seeing middens, but I'm seeing them over here. So I'm going to give it credit. Middens present, cast present. Soil smell score 2 low, 2.5 high. And then the other things that we did when we were doing these field assessments, we did penetrometer readings. You can approximate that by taking a flag, one of those staking flags you get at the hardware store about this long, and just push it down into the soil. And if it pushes down easily, then that's good. And if it bends, try another spot. You might have hit a rock. But if no matter where you try, you cannot push that thing down 6 inches, 12 inches, 18 inches, then you know you're hitting various layers of compaction. So I don't have a penetrometer, so we're not doing that one. That's a tool that measures how many pounds per square inch you're putting to drive it down into the soil. And we're not going to worry about doing an infiltration test because I don't have any good coffee cans at home anymore. My husband buys all that blasted bag stuff. So if you wanted to do an infiltration test, you just take a can that is sturdy enough to be able to be pounded down into the soil, put a board across the top, bang it down, have your phone or your stopwatch handy. We put a little bit of plastic wrap down on the soil so that you can protect the surface. You firm the soil to the inside and the outside of the can, and then you add an amount of water that you have measured. So what we typically did was we'd have a 6-inch brass ring that we pounded down 6 inches by 6 inches. We pounded down 3 inches. And then we had 744 milliliters of water, which would be the equivalent of an inch of precipitation sitting in that area. And then we'd see how long it took to disappear. On soil like this, it probably wouldn't take very long. The difficulty of doing infiltration tests is if you happen to hit an earthworm tunnel, then everything disappears really fast. If there's a soil crack, everything disappears really fast. If you didn't get the soil sealed to the inside edge, then everything disappears really fast. Or you could have a rock down there and not know it, and it just takes forever. So that one is one that you have to, if you're curious about it, you do it in as many places as you care to, and then take an average. And you try to avoid obvious things, earthworms, tunnels, soil cracks, being right next to a plant. Being in between 2 plants is okay, but right next to one you try to avoid. And then you can take a look at that. But the quick and easy way of determining whether you've got good infiltration in your soil is just to look outside when it rains. If the water is going down into the soil, it's going down into the soil. If it's ponding up on the surface, you have a problem. Either your soil is completely saturated or it's not allowing the water to come in. So this is kind of a quick and dirty soil health assessment of this soil.